All right, good morning, everybody. I want to thank you all for coming, and especially this crowd for coming out early to the start of our symposium. First, there's still plenty of seats up in the front, so for those of you that want to move up, this is your chance to do so. We're not going to charge you any extra for sitting up here at the front. It is a real pleasure. This is our third symposium, and it's been quite an exciting year if we think about the things that have come down the pike for the center in terms of the fellowships that have been funded, the projects that have been funded, the larger initiatives that Mark will talk about briefly. We're really sort of in the real thick of things as we grow and as we continue to expand. One of the things that has been particularly gratifying to see is that when we started the center, we said that one of the foci would be to think about the information age of genomics and the opportunities that arise as we bring ever higher resolution data to bear on problems of biological, anthropological, and medical importance. And that, I think, has been fully realized as we've seen the real shift both at a national level, but also on campus in terms of the big data initiatives that have started in large part from the discussions that began to start the center. And there's probably no better indicator than that of having our keynote speaker today, Gene Myers, will tell you a bit more about in a few moments, as well as our co-presenters and for the morning session, DNA Nexus, who had invited Gene to speak, and we were able to borrow him for a first few hours, so we want to thank them. And as we look out, you'll see in the 2 days of the symposium, so if you remember, our first symposium was an afternoon, our symposium last year was a full day, and now we're sort of into a two-day symposium, the sort of interweaving of these elements that have to do with bringing both computation and large-scale technologies to bear on important biological problems. And this sort of first session that we're starting first with Gene's keynote and then a series of talks from fellows and faculty sort of highlight that theme, the importance of both the computational and the experimental, and how you really can't tackle many of these questions without bringing them together. One of the things that in particular we're pleased to announce is that we've now started sort of full-fledged on a couple of flagship projects for the center. One of these has to do with large-scale Y chromosome sequencing. So Mark was able to get a very generous donation for a project that is focused on Y chromosome sequencing, and we've leveraged that with center funds, and will hopefully, by the end of the year, produce the largest set of fully sequenced Y chromosomes to date. We're targeting, on the order of a few thousand Y chromosomes to be sequenced by the end of the year, and making that resource available to the community. It's really sort of the beginning of putting together a much larger reference database on human genetic variation. The second large theme project that we've begun is a collaboration with multiple sites on understanding African diversity as it relates to the transatlantic slave trade, and that's really become sort of a wonderful theme that has resonated through several of the funded grants in the project. And so we encourage you to keep your ears to the ground to that. We invite you to participate, and also to say that as we think about going forward as a center, having a few flagship projects on which we anchor the central themes is something that to us is very important. We don't want these just to come from the directors or from the executive committee. We really encourage you to come to us, and we'd love to think more together as a center in terms of what these flagship projects should look like. Mark, I don't know if there's something you'd like to add there, or if I've... So right, and the idea is that these really are flagship projects that are open to all. So for those of you who are interested either on Y chromosome sequencing, large-scale human genetic variation, or the transatlantic and now even the Indian Ocean slave trade, please come talk to us because we'd love to involve you in those projects. Okay, without further ado, I'd now like to introduce our keynote speaker. When I think of Gene Myers, I really think of him as quite a disruptive force, if that's okay to say. If you think about what the world looked like, certainly when I started graduate school, I remember going to meet with Lewontin, who'd been my undergraduate mentor and ended up on my committee, and telling Dick I wanted to be a theoretical population geneticist, and Lewontin looking at me and telling me that's possibly the worst idea I've ever heard. You know, you're not that good at math, Carlos, and this has become such a difficult field that you basically have to be a probabilist to make any headway, and every good place already has a theorist. Who on earth is going to want two people who are analyzing genetic variation and not generating their own data? That's just kind of crazy talk. I said, well, you know, I'm not that good at experiments, so I better work on the theory side and better work on the statistical analysis side. And this was like 96, 97. And I finished my thesis in 2001. And Clinton and Blair had just announced the completion of the draft project of the human genome, which wasn't slated to be finished for another 10 years, right? And part of the reason that that project got accelerated is due to this guy. He and the development of the theory for really assembly and large-scale assembly from read sequence data revolutionized the world. He went to work for a small company called Celera that was racing the government. In some ways, it was a very clever idea for ABI to spin that out. They sold sequencing machines to both sides, and so both the federal project, the international project, and Celera went on to tie in their sequencing of the human genome. And as I said, it was really sort of the technology and ideas that Gene had developed that moved that forward. From there, not happy to stay in one place and rest on his laurels, he moved to really switch research directions and begin to work on the next frontier of questions involving imaging and microscopy as part of the HHMI Janala, I can never get the name right, Janala farm, which, as you know, was a very interesting experiment that they spun out, bringing together a set of investigators with small labs to tackle very interesting problems. In many ways, for those of us who are scraping around for federal funding, having a position like that would seem, boy, why would you ever give that up? But as I said, not one to rest on his laurels. He then upturned again and is now the director of a Max Planck doing systems biology, and we were just having a wonderful discussion over breakfast of all the intermingling of things that he can now do as a director of a Max Planck, and it's really quite exciting to see. I always talk about models of success and thinking out, okay, who do you want to be in 10 years or 20 years or 30 years? And Gene is really sort of at the top of that list of somebody who's woven in and out of industry, in and out of academia, and now in sort of a partnership research program, and somebody who's always really been, not only months, but years ahead of the field. So it's a real pleasure to have you here. Thank you for fitting us into your schedule. And ladies and gentlemen, Gene Myers.
Thank you. Is my lab on? Can you hear me? Yeah, great. In a way, this talk is back to the future for me. As Carlos said, after finishing sequencing the human genome, I felt like in 2002 we had done a pretty good job of reconstructing it. But at that point, what I saw was that there was this push to reduce the cost, and everything went towards basically short read sequencing. And the original goal of actually producing beautiful reference quality genomes, right, perfect genomes. So imagine getting your genome correct from telomere to telomere, okay, and having every base be correct, right? I mean, that's the standard, right? And we were instead of moving towards that after the Celera days, we actually moved away from it dramatically. And so, in fact, and it's only been in the last couple of years that I've come back to it, and I'm going to explain why. But basically, as Carlos said, what happened is I developed another vision, in part I started with Stuart Kim, and we thought about worms, and the idea is to really basically be able to digitalize, build digital atlases of cells, tissues, and organisms, and to be able to annotate them with molecular information. And so to really, in some sense, quantitate this idea of cells to tissues. And here are some examples of some of the things that, there's a fly wing developing, and we build models. These are the kinds of things that my group has really been doing since about 2003, 2004. But what I'm going to talk about today is assembly. So what I'm going to do is here's the kind of the organization of the talk. And I apologize, it's a tech talk. I'm not going to talk about human population genetics. But it's a tech talk that I hope that what you'll see is that coming down the pike is this vision of getting a genome perfect from telomere to telomere is really going to happen, okay? And eventually, I mean, you don't have to be a futurist, eventually at a price point where it's going to basically transform the work that you do, even as a human population geneticist or as an evolutionary theorist, right? I mean, in some sense, the genome is the source of all the information and all the analysis that we do. And wouldn't it be wonderful if it was perfect, right? I mean, because so many of the things that we deal with every day are fully imperfect and full of caveats, right? So I'm basically a technologist, and my current goal, what I'm very excited about, is this idea that we can perhaps do perfect assembly. So I'm going to talk about, why I think perfect assembly is back on the table. And then I'm going to talk a little bit about the work that I've been doing in that regard. And I don't know how much time I'll spend on it. It depends upon, how I see you reacting to what I'm talking about. I get very excited about the details, but I certainly can appreciate that you might not share my enthusiasm for those. But basically what happened a couple of years ago is that Mike Hunkapiller at PacBio gave me a call and said, hey, look, the instrument's going to start producing 5,000, 6,000 base pair reads on a regular basis. And in particular, and so now this machine, it's not that I'm wedded to this particular machine. What I'm wedded to is the characteristics of this machine. I'll say that I'm technology agnostic. If there were another machine like this one, I'd use it, especially if it was cheaper, okay? But this machine has some very unusual characteristics, and in fact, it's the first machine. So it spits out very long reads. The reads are now over 10,000 base pairs on average coming out of the standard libraries with the P6 C4 kit. And you also know that it has very high error rates. That the error rate is 10 to 15%, okay? And they're mostly insertions. But here's the key, which is that there's some very important caveats, which is that the sequencing errors are random at any given point. In other words, there's no predisposition for there to be an error at a particular point in a particular sequence. This has not been true of any previous technology. Illumina, Sanger, you name it. Okay, about 1 every 10,000 base pairs, those technologies will make the same mistake over and over and over again, right? Okay, so you'll never know what that particular base is because the machine will always be making the same mistake. This machine is completely random. It's basically, if you look at its noise, it's basically thermodynamic noise, which is a perfect random number generator. The other thing that's incredible about this machine is because it's a single molecule technology and because of the way the preps are working, there's almost basically no bias in what it is that you end up sequencing. So in other words, if you reach in and you grab a piece of DNA and say, I'm going to sequence it, you reach in and you grab every piece with equal probability. Whereas, for example, with Illumina, there's very strong CG bias, right? If you have a strong CG region, typically it doesn't PCR very well, and so you don't see it, right? So you end up with holes in your genome, right, due to the sequencing bias. Whereas this machine, basically, as far as I can tell, seems to sequence everything. The centromere's in there, the telomere's in there, everything's in there, okay? At relatively, basically, Poisson distributions, right? And so this is in some sense a mathematician's dream, an informatician's dream, because now I actually have the underlying conditions that I would like to use theoretically, right? Which is that errors are random and the sampling is Poisson, right? Truly Poisson, right? Which are the conditions, right, that make the whole theory about being able to do assembly, okay, available, and in fact applies that near perfect assembly is possible. So the first part of the talk is probably the important part, and then you can kind of go back to your laptops or whatever it is you want to do, unless you're excited about the details. But I want to explain to you why I think that perfect assembly is now possible, okay? And so I tweeted, just for fun, I tweeted 140 characters theorem last year, just I was new to Twitter and I was having fun. But basically what this theorem says is it says that if the sampling is Poisson, if the errors are random, and if the reads are long enough to resolve repeats, which is a bit kind of vague, then perfect assembly is possible. And I've been very excited about this ever since. So let's take a look at a couple of let's just, for those of you that aren't familiar with the old theory, this is actually very old theory. What about the fact that the read sampling is Poisson? What it means basically is you take any level of coverage, 10, 20, 30, and basically if you do enough sequencing, what it says is that it says you'll end up covering the entire genome at that depth. It's just a matter of persistence, right? But you will eventually obtain it. You will get everything at the desired level of coverage, right? So this is basically Lander and Waterman from 1988, okay? So with enough sequencing, it guarantees that any minimum coverage depth is achievable. You will eventually cover every part of your genome 20-fold if you do enough oversampling, if you do 100x or whatever it takes. I won't say exactly what that number is, but that's what the implication is, right? Without that condition, right, it doesn't matter how much sequencing you do. If the region's CG rich, you're not going to get it, right, with some technologies, okay? What about the error read is random? Well, everybody's used to this Q-score, which is a logarithmic score, and basically a very old theory by Waterman and Churchill from 1989 says that if you pile the sequences up high enough, okay, and regardless of what their error rate is, you vote, and you know that the underlying error, that the errors are random, that the consensus that you get out of voting gets better and better, becomes more and more close to the true answer, and in fact it converges to perfection geometrically. Okay, so what it says is that the quality of the reconstruction you get, right, increases in terms of Q-score. It increases linear in the depth at which I have it. Okay, it's converging exponentially. And so what that means is that it says that okay, if you do enough sequencing, then you're guaranteed to cover everything deeply enough. And if everything's covered deeply enough, then you're going to get a certain level of accuracy, right? So in some sense, this is the importance of those two important conditions. It's also the case, and I didn't really address it in that little theorem, but I want to talk a little bit about what kind of repeats can you resolve and how does length participate in that. So if you have a sequencing read, and that read spans a repetitive element, so suppose you have some 7,000 base pair full-length retrotransposon that occurs in your genome. If that 7,000 base pair retrotransposon is in the middle of a read, and on either side the read has at least, say, 1,000 base pairs of unique sequence, then you can resolve that repeat. It's not an issue for assembly, and the answer is very simple. If you think about it this way, for example, suppose this is unique sequence, kind of to the left of the repeat. Here's the repetitive element. This blue element is this kind of this big box. So there's 100 copies of this retrotransposon, say, in the genome. So there'll be 100 ways in and basically 100 ways out, right? And the problem is, how do you get from one side to the other? Well, if the read has unique flanks, right, then it clearly tells you how to go in and how to come out, right? And that's all there is to it. So and this is way back from the Celera days. And basically it says with enough sequencing, every repeat of length R minus 2F will be resolved, where basically R is the read length and F is the kind of the amount of flank that you want. And for this technology, it's about 1,000 base pairs is more than enough to basically say, yeah, I'm definitely here, I'm definitely there. And R percent is a little bit complicated, but say R25 is the 25th percentile read length, okay? Which, for example, for this instrument would be about 15,000 base pairs, okay? And you can pick any percentile you want. You can pick R50, R25, but I picked 25 because that's a reasonable level. Because at R25, if I do enough sequencing, I'm going to have enough R25 length reads that I'm going to basically be sure to span every repeat. And so basically, R25 for the technology is currently 15,000. So 15,000 minus 2 times 1,000 is 13,000 base pairs. So basically what it says is with this technology, as it currently stands, basically any repetitive element that's isolated in the genome, that's interspersed, and less than 13,000 base pairs, no problem, right? Absolutely not a problem, okay? Gets assembled. And you'll see this later. So, but even longer repetitive elements are solvable. So here's a big one. Okay, so here's a big hairy one, okay? It's tens of thousands of base pairs long. And here kind of the unique ways in, so there's 100 ways in that are kind of coming from unique flanks, and then they enter this no man's land, and then they're supposed to come out into unique areas where you, again, are very sure that you have, you know what you're putting together. So the question is, how do you get across this? Well, if there's enough microheterogeneity, okay, inside that region, then you can even resolve it, okay? And the interesting thing is nobody's written a piece of code to do this, but it's clear, right, conceptually that can be done. So imagine that those the spots that I just put up are regions where there's variation between the repeat copies, and and I'm just using color to denote variation. So there's the white base, and there's the red base, there's the white base, the yellow base. And the point is that if there's enough variation, okay, then you can basically split the reads into classes according to their vector of variation. And if you then have reads that basically overlap each other, and the overlaps are unique in terms of the heterogeneity fingerprint that they have, then you can basically get from one side to the other. So the question is, how much of that do you need? And I'm just going to pull some numbers. So from an old result that I did in 2002 at the Japanese GIW conference, basically what I showed that in the background of 15%, if you had about 5 microheterogeneities, that was enough to resolve 2 copies. You could pull 2 copies apart with 5 microheads in a background context of 15% noise. And that's basically an empirical result. Which means that if I want, say, a sequence of 66%, just to give a sample number of overlapping reads, then it means that I could sustain about 1 heterogeneity every 2 kb for P6C4. So that's pretty good, right? I mean, if you have 1 microhead every 2 kb on average, okay, through that region, you're going to resolve it. So what it means really is really only the things that are like 99.99, okay, and are 100K long are not gonna fall. Now, this begs still the question of nobody's actually built an assembler that does this well, which I actually find really surprising, okay, given that when I left the field 10 years ago, it was clear that this is what you needed to do. And I think that, honestly, if I could be a little bit biased, I could say that I think too much of the work has focused on speed and efficiency and not on correctness. Okay, because I think for this problem, you want it to be right, okay? I don't care how fast it happens, or how many CPU hours it takes, or how much memory it needs. What I want is I want it right, because I only need to do it once, right? When that one throw of the dice is over, I want it to be beautiful. Okay, so why people are competing about the speed, okay, secondary to being correct. The real issue here is being correct. So what I've hopefully kind of given you a feeling for is that with these new technologies, and perhaps the nanopore technologies will deliver data like this. And if they do, and they're cheaper, then I'm going to switch to it, okay, for sure. But you can see that there's certainly a trend, and it's very exciting to me that we're kind of moving back towards long-read technologies. Yeah, it's more expensive than Illumina, but it's also 10 years behind the technology curve. Give them 5 years, and it's not going to cost much. Again, you do not have to be a futurist. The cost of long-read sequencing will go down. It's already going down by a factor of 2 every year, okay? So at some point, right, this is the way we'll do it. And the crux then, but then the real crux is, and the thing that I didn't really address is, how much sequencing do you need, right? And how do you deal with that error rate, right? I argued in a principle, but so then what it really comes down to is that you have to build an assembler that can deal with a 10 to 15% error rate, which nobody has really been focused on in the community. And the other is really how much of this data do you need in order to get the characteristics that I'm talking about being sufficiently accurate, about having everything covered. Okay. But what I want to point out is that just kind of where we're at today, and I certainly haven't finished building the assembler I want to build, but just currently where we're at is that the current assemblies with these kinds of data sets are already competitive. For example, D. melanogaster is probably the best sequenced, okay, and best vetted genome in the world, right? Release 6. Okay, those are the release 6 statistics there. And you can see a PacBio assembly out of the box with the current software, which is by no means optimal, okay, is basically competitive, right? I mean, there's a slight difference in the numbers, but look, they're almost the same, right? The N50s, the maximum contig, I mean, the level of continuity, the number of base pairs delivered. In fact, the PacBio delivers more base pairs because it's delivering all the centromere, which isn't really assembled in that, right? But think about that. I mean, there's been 10 years of finishing on that genome, right? It was done in like 2000 at Celera, right? And then they've been finishing it for 14 years, spending a lot of money doing things. One PacBio run, I mean, I think it's like 8 SMRT cells, okay, which means it's not even really that expensive, like 10 grand. Okay, and you run the assembler and you get something that supersedes or equals all that work. okay? I mean, that's what's happening, right? That's my main point, right? You guys get it, right? I can probably stop at this point, right? Okay, yeah, I mean, that's the take-home message. Yeah, I should keep going, all right, okay. But, you know, and this is on a fairly small genome. On a bigger genome, you know, we need a better assembly, but still, I mean, HG38, okay, again, another 13 years of finishing effort, millions of dollars spent, okay, and you look at a PacBio 84X assembly of Craig's genome, and, I mean, okay, it's a little bit weaker, okay, but not bad, right, not bad at all. And in fact, if you do something like run like BioNano and get a physical map, okay, which basically the BioNano map has about the same stats as the PacBio assembly, okay, so it actually doesn't really improve on it. But if you combine the two, suddenly you have stats that are just as good as HG38. Okay, it's pretty impressive, right? I mean, again, right out of the box, right? A month of sequencing. Okay, a little too expensive, right, to do it for human variation today, but in 5 years, I'm not so sure. Okay, and here's another kind of view of kind of like what the assembly looks like on human. And I was working with Jason Chen at the company very closely. Okay, so now I guess what I'll say is the rest of it basically is about the part that's fun for me, which is how to build this assembler. And in a way, I mean, I'm going to give some clues about how I think you need to build such an assembler. So first of all, let me kind of describe kind of what's out there. Okay, so for this data, basically, an assembler is not a kind of a monolithic tool, right? It's not like BLAST. You know, you give it a sequence and database, and it's out. It's really, I mean, technically, you're giving a collection of sequences, but there's like really a lot of steps that you want to perform. And basically, the general idea in these paradigms is it's the overlap layout consensus paradigm, which I'm guilty of like 20 years ago. But the idea is you basically take all the reads, you overlap them to see how they might be able to go together. It's like taking all the pieces of the jigsaw puzzle and saying, could I fit with you, okay? And so you do that. And that's usually the very CPU intensive part because you have to do a big all against all comparison. And then for the PacBio data, Jason Chin came up with a very interesting idea, which is I take a read, I look at every read that aligns to it, and I use these reads to develop a consensus of what this read should be. And what that does is that's very interesting. It takes you from a read that has a 15% error rate to a reconstruction that has a 1% error rate. Okay, although there are some caveats, because if you don't do the right voting and you let repetitive copies vote, then you don't necessarily have the consensus. But generally speaking, what it does is it cleans up the data so that it's 99%. And then what you do is you run a traditional Celera-style assembler. You overlap at 95%, which is what we did at Celera. You do the assembly, the string graph idea, and then you produce a consensus sequence, okay? So all you did was, so the key thing was to kind of deal with the error rate. And so the first assembler that came out by Jason was HGAP, where these were the components that he used, one of them being the old Celera assembler. And then more recently, he's come up with Falcon, where it uses these components, okay? And basically what's happened is he's slotted in my overlapper because it's much faster, okay? And so here are the contributors to these components. But the idea is that today it's a little bit complex to use this particular data because the software base is not mature. And so basically, if you really want to go out there and try to affect an assembly with PacBio data, you've got to kind of strap on, right? That you're going to have to kind of figure out a bunch of modules that have been developed by different groups and figure out how to string it together. So you need a real bioinformatician, right, on your team in order to use this thing. Okay. So as you can see, I mean, I got into the game about 1.5 years ago, and I've managed to build this component. Okay. And the problem, though, is to build beautiful components. Each one of these components is a big job in and of itself to build it beautifully. I'd say at least a man-year for each one. And so I've been basically, I've been a one-man team so far. I do have a sequencing staff, and I run a couple of PacBios, but the back end has been me, okay? Because everybody else in my group is thinking about images, okay, and vision and tissues. So basically my idea is I'm going to basically build the best module I can and release it as soon as it's valuable to you. So my goal is to basically move through this entire chart and basically at some point dominate the world and have all of the bubbles be orange. Okay? And at that point then you will have the perfect assembler, right? and nirvana will ensue. So what I am doing though is I finally said, look, this is serious enough for me. I'm hiring a couple of postdocs, great postdocs. They're coming on May 1st, and I'm hoping that we'll hopefully there'll be more speed once that team joins me. So and I'm very committed to kind of being open source, so you can go get all this stuff. I have a real kind of funky blog. There are no opinions there. There's just code. And basically, effectively, a kind of informal documentation style for code as long as informal code. And in particular, from there you can also get a link to GitHub where you can download any of the released modules, okay? So just remember the Dazzler blog and you'll get everything. So let me tell you about some of the modules that I built and released. Some of them are very basic. So one of them is if you for example, the haplotypic mole that Evan Eichler wanted sequenced, they did a 50x sequencing of a human haplotypic mole. The data set occupied 2 terabytes. It's in what are called .bax.h5 files, and that's rather large, right? So the first thing I did is I built a tool that basically goes into that .bax.h5 file and extracts just what you really want, which is the sequence data and the quality values for the sequence data. And it gets rid of all the other stuff, pulse, height, temperature. I'm never going to use that, I don't think, okay? So that already gives me a factor of 4, okay? And then what I did is I built a couple of lossless compressors that give me about another factor of 3.5. So I take that data, and without losing anything, I just do compression, okay? And compress them into small forms. And I can decompress those anytime I want, okay? And so that gives me about a factor of 13.5. So that 2 terabyte data set goes to 150 gigabases, and I have everything that I want, right? So I basically have every bit of data that I've ever sequenced with my 2 PacBio machines over the last 2 years sitting in 2 terabytes, okay, on my machine. 2 terabytes, I mean, it's equal to this data set because I've compressed everything, okay? I have a 30x axolotl data set, a 50x planarian data set. I have quite a few data sets, okay? So what I generally do then, and this is a module, it's available. You can get this code, it works, it's been debugged. Okay, so if you're working with this data, I would strongly suggest you compress your data, roll your .bax.h5 to tape, put it on tape, because you may want it back. I don't know why, but you may. Okay, so put it on tape just to be sure. Okay, it's cheap, right? And just keep the compressed files on data. Okay, so the next tool then is really more of an infrastructure module. And it's kind of required for everything that goes downstream, but basically I needed to organize the data. I needed to create some kind of database. In particular, I needed to at least be able to get at the reads and the quality values in a random access fashion. So instead of compressing them into distinct files, which you can do, I actually compress them and put them into a database. Okay? So now the database contains the compressed data, but what's cool about this database is that it gives me direct access programmatic database. So this is more for a programmer. But if you're a programmer and you want to use this data, if you're willing to kind of take a look at my libraries, right? There's code in there that says, go get me the 23rd read, go get me the quality values for the 25th read, right? And it just loads it up for you. So if you want to build code around this, I mean, if you want to compete with me, great. Take this, but this will give you the initial infrastructure for this, okay? And it's all been done, and it's been done in a very nice way. But it has some other things. Is that, for one thing, it's completely lossless, which is that if you put the stuff into the database, okay, and you ever want to get back your FASTA and equivalent files, there's commands that say undo it, and it'll give it back to you. Okay, it'll give you back your .fasta files and equivalent files. So don't worry, you never lose anything. Okay, I felt that was very important psychologically for users, right? That they knew that their data was still there. Okay, and so it's completely reversible. Okay, it's not a one-way street. The other cool thing is that you can build this database up as you're going. So as data is coming off the machine, you can just keep adding FASTAs and quivas to it. It'll build it incrementally. Another nice thing for the programmers or for a developer, if you're a developer, is that there's this idea of tracks. You can kind of keep any kind of metadata that you want about a read associated with it, like low quality intervals or things like that. And probably most importantly is that these computes take a lot of time, and this notion of a database intrinsically supports the idea of a partitioning into blocks that allows you to then do, large-scale job parallel computations on an HPC cluster. So it immediately supports all of those things, and I think it's if you take a look at it, you might enjoy it. So the other two modules that I built are the Deliner module, and the Deliner module basically is incredibly fast. Okay, because I did feel it was for the human haplotypic mole with the existing tool, it took 400,000 CPU hours, okay, to do the overlap phase. And I felt that despite my comments about you want to get it right versus you want to get it wrong, that's enough CPU hours that it's daunting to most of us, right? It's even daunting to me. I mean, I'm thinking, my God, it's, you know. Fortunately, thanks to DNA Nexus and stuff, that kind of stuff has now been made easy for you, okay? But even still, I worked at it and I got it down to 15,000 CPU hours. So I got about a 25 to 30x speed up. I was going to tell you all about it, but you know, I'm not going to tell you all about it, okay? Because I think it's this kind of stuff that I love, but I think it's enough to say that it's fast. The other module that I developed is a scrubbing module, and now it actually starts to get interesting, okay? Carlos, when should I finish? 10? Should I finish at 10? 10 is okay, so 15 more minutes? Okay, I just don't want to, you know, I'm going to have to excise some slides, which is typical for me. So let me tell you about scrubbing, okay? All right, so what do I mean by scrubbing? Okay, so the idea of scrubbing all began with the following thing. So I had left Celera, it was like 2004 or 5, and I'd kind of done my final work on how to build a string graph, how to do the assembly. And basically the nice thing about assembly is that what you really want in some sense is if there are large repetitive elements in the genome that you can't resolve with respect to the read length, then what you'd like to do is get out a graph in which you at least know that the tour of this graph is the correct assembly. So here's actually a synthetic assembly of CEHG, which is a 1.8 megabase pair bacterial genome. I took the real genome, this was 2005, I generated 700 base pair Sanger reads with no mate pairs at 10x, and I said, okay, I gave it to my assembler, and I got this beautiful graph back, which says that basically if I start here, and I tour this graph, and the edges on this graph are labeled with sequence, okay, big hunks of sequence. I want to get from here to the end, and these red edges, which correspond to repetitive elements, I even know they're repetitive, and I can tell you how many copies there are. Like this guy occurs twice, this guy occurs 3 times. I need to find a tour through there that exactly respects the copy number. And if I find a tour from here to here that does that, then that's an assembly of the genome. It turns out that there are 72 possibilities, and 7 PCR reactions would resolve it. Okay? But that, in some sense, is the best information I could get with 700 base pair reads, given that these red stretches represent repeats that are much bigger than 700 base pairs. And in some sense, what I'm saying these days is that is the goal of assembly. The goal of assembly, I think everybody's used to this idea now that the output of assembly is a graph, right? People are talking about graph genomes, okay? Well, we knew this back in the 90s, okay, but it just wasn't popular, okay? And really the statement is that you want to produce the graph that is as most resolved as possible, such that some tour through that is your answer. Now the interesting thing is that I took those 700 base pair synthetic reads in which I had basically sampled the underlying genome at 10x completely randomly, and I had introduced 5% error into all the reads, so I kind of introduced mutations, and I assembled and I got this beautiful result. Okay. But then when I took the real data set, I went to the NCBI and I downloaded the real data set and I gave it to my tool. I got a crap result. I got this big kind of hairball graph. Sorry, I don't have it to show you. Should have kept it. Okay, but the program couldn't even draw it. It was so awful. It had so many branches and hairballs in it that it just looked like crap. And I was like, well, what the heck is the difference, right? I mean, oh man, I thought I built this beautiful code. I know that it should build a string graph, right? But I take this real data set and I'm getting this crappy looking thing, right? What's going on? Well, what were the assumptions, right, of the simulated data? The assumptions of the simulated data were that the reads were contiguous stretches of the underlying genome, and that the reads had a maximum of 5% error, right? I didn't introduce more than 5% error in any of them. So what has to be true of the real data? What has to be wrong with it? Well, one of those two conditions has to be false, right? It has to either be the case that the reads are not contiguous stretches in the genome. How does that happen? Chimers, right? Chimeric constructs. You forget to remove the adapter on one end or another, that'll blow that thing, okay, and you can go on and think about other combinations. Oh, foreign DNA, okay? DNA is not from the organism, okay? Or that the read is noisier than 5%. Well, how did that happen? Well, you know, like Fred, unfortunately, isn't perfect, okay? Everybody assumes that Fred is actually generating a true quality value. What it's doing is it's generating an estimate of the quality value based on a machine learning technique, right? And it can be god-awful wrong on some reads, okay? Fred could say this read is 98% accurate. In fact, it's a 90% read. Okay? It doesn't happen very often, but it does happen, okay? So those are the things that are wrong, right? So, but in a way, there's kind of good news here, right? Because we know that if those two conditions are met, then assembly, in some sense, the part about putting the reads together is a solved problem. The original string graph theory, right, the chordal graph collapsing from, you know, my 1996 paper, exactly gives you what you want. It's a perfect assembler. There's nothing wrong with it. It does the job. So it means that we don't actually have to work on the part that decides how to put the pieces together. What we need to work on is the quality of the pieces that go into the problem. Okay, right? Duh. Okay, sorry, probably it's obvious, okay? I want to make sure about that. Okay, so the real data problems are the chimeric reads, the contaminants, the unclipped. And basically what I would say is that the conclusion is that this, I call this scrubbing, to get rid of all this stuff. I'm going to scrub the data so it's clean, okay? It's underappreciated. Other people that have been working on this problem for the last 10 years have not been paying attention to this, because this is the major impediment, okay, to great assemblies. Well, that and the reads are too short, but I can't do anything about that. Okay, I didn't take it in that direction. So what is a scrubber? A scrubber is basically, the goal of a scrubber is to remove as little of the data as possible. So you're going to edit the data, you're going to break chimers, you're going to remove untrimmed data. So you're going to do some edits, right, to the reads. And you want to do that so that you throw away as little as data as possible, such that when you're done, every edited read is a contiguous stretch of the underlying genome, and every part of an edited read is of a reasonable quality, which for PacBio is better than 20%. Okay. All right, so, and if you do that, then you're going to get that. Okay, so what I did is instead of working on so the next thing I did is after building an overlapper, was instead of working on the string graph, whose algorithm I'm completely happy with, what I spent the last almost year doing is basically looking at how to scrub the data. So I'm going to skip over finding local alignments, very, very geeky stuff, okay, but it's published and you can find a paper on it at the workshop on algorithms for bioinformatics, or WABI. And I'm going to go directly back to intrinsic quality values and scrubbing. Okay. So the PacBio reads introduce an interesting problem, okay, because most of us are used to kind of the shorter read era. When you think about a sequencing read, okay, so this is the read from 5' to 3' along the axis, and you think about the error rate, right? Well, typically what you see is the profile has always been in the past that, you know, error rate usually starts a little bit high, it quickly settles down to some nice level, 0.5% for Illumina, right? And then you get to some point where the read starts to break down, like 150, 250 for Illumina, and it starts to take off again, right? For Sanger, that was about 800 base pairs, right? So you have this kind of very simple error profile, which kind of predictably would say, hey, look, if I cut the first 50 base pairs and I cut at some point, everything should be fine. But what about PacBio data? PacBio data is not like that at all, right? So the reads can be 20,000, 50,000. We've seen 70,000 base pair reads, okay? And what does the error profile look like? Well, it does tend to start high, okay? So some things have remained the same, okay? It settles down to its mean rate, which is like 12 to 15%, okay? It's going along, okay? Suddenly it gets a little bit hotter for some reason or another. The next 2,000, 3,000 base pairs, it's running 18%. Then it suddenly hits a sweet spot. It's running 8% for another 1,000 base pairs. And then suddenly, for 100 base pairs, it goes completely apeshit, okay? And just generates some crap, okay? Just generates A. Okay, and sometimes it's not that easy. If it's homopolymer stretch, I'd get them all. And then it recovers. And it's back down there at 12%. It's going along. And then suddenly, you'll get to a region, and it'll go nuts again. Okay, I'm not sure what's going on. I think it may be that the DNA is folding back over the Paul binding site or something, and kind of inherently. But basically, the machine can't really tell that something's wrong, and so it's spitting out crap. Okay, and it's doing it for 3,000 base pairs, and it's not a homopolymer run. Okay, it's guessing things. It's basically observing noise in the well, okay, and spitting out a random sequence for 3,000 base pairs, and then it recovers, okay, and goes on again, okay. So this, you have to think about this kind of a read, right? What are you going to do with that kind of a read, right? How are you going to make sure that it's all of a uniform quality? How do you even know what the quality of this data is? And the really sad thing is that you can't get this from the QVs that come out of the instrument, okay? And it's not to jam the guys at PacBio. They built a beautiful support vector discriminator just like Phil did, okay? They looked at all of the profiles of the machine, but the point of the matter is that you're talking about a single molecule technology, right? And it turns out that there just isn't, I think intrinsically there is just no way to know, okay, that the thing has gone south. Okay, there's just no way to know, or very little way to know. The quality values, I wouldn't say the quality values that they produce are completely uninformative, but they're not enough to bet the bank. And that, like in that 3,000 base pair stretch, the quality values will say, yeah, it's not so good, but it won't say it's completely out to lunch. It won't say it's random. It'll say, oh, it's 20%, okay, which is definitely not what it is. Okay, so the biggest problem with this technology is you have no idea how good the read is, okay? It's a real problem, right? Because I said it's essential. So what I do is I do this idea of taking a read and comparing it to all the other reads. So I need you to understand this thing. So the gold bar up here, the yellow bar, is a read. It's read 106, okay? And all the white stretches are the intervals of other reads that align with that read. Okay, so for example, down here in the corner, you'll see here's the read. Here's another read B, okay? And this part of B aligns with A, okay, this interval. And then the rest of B doesn't align with A, which I'm going to kind of denote by a brown bar. The alignment stopped, okay, at this point. So, and that'll be drawn in the pilogram like this. It'll be white for the interval that overlaps, and then a brown tip to indicate I stopped aligning with you, right? Okay? So you can see, there you go. Those are all the other reads in the data set that align with A, okay? And how they align with A, and whether or not they're complete alignments, or whether they are aligned and then the alignment stops at some point in the B read. Does everybody get it? Important you get this one, okay? So I call that a pilogram, okay? Because it's a pile of reads, right, that align to A, okay? So this would be kind of like a normal pilogram, okay? What about this pilogram here? So they're already really informative, okay? So, well, nobody aligns with the first, 500 base pairs of that guy, right? That suggests that probably you want to trim that guy, right? Here's another one. Gee, nobody aligns through this stretch. Something's going on there. It's either a chimer, it's a chimeric break, there's some unremoved adapter as a possibility, or it's just that the read just really sucks right there. It's really low quality, right? So nobody aligns with it because it's in one of those zones where it's spitting out crap, okay? And then here's another interesting pattern, right? Look at all those guys down there. That's a repeat, right? That's some kind of repetitive element in the genome. Right? So you can even find repetitive elements, okay? So this is very cool, right? This kind of immediately kind of psyched me because I'm like, oh, okay, cool. I got all these patterns, right? And I'm a pattern matching guy, so kind of, okay. But still, I'd like to understand quality. So that kind of gives me a feeling for what kind of globally might be going on. But I'd really like to understand whether bases are good or bad, okay, in a given region. So how am I going to get that? I'm going to get that from the pile. I'm going to do it in the following way. So I'm going to go just for a moment into a detail about alignment. So I hope you guys will forgive me, but basically the aligner reports that, for example, the interval from AB to AE of read A aligns with the interval from BB to BE of B, right? So this part of A and this part of B align, okay? In the edit graph, which is what kind of the computer geeks like to use, okay, it corresponds to a path from this vertex to this vertex, where this is kind of the A axis, and here's the interval from AB to AE, and then here's the B axis, and it corresponds to the interval from BB to BE, and it corresponds to kind of the alignment corresponds to a path in this model. And generally, so the problem is I could give you that interval, right? That's 4 integers, right? Or 6 integers, which 2 reads and the beginning and end intervals, right? So 6 integers encodes the overlap. That millions of overlaps come out, right? When you do the human genome, you get like 2 billion overlaps, okay? So how are you going to code all those things, okay? And you have basically two bad options, according to current theory. One is that you just encode all of the edits. You say, oh, there's an insertion there, there's a deletion there, there's a substitution. You do like BAM, right? That totally sucks, okay? I mean, you would basically be blown out of the water, okay? Because, I mean, you're talking about an alignment of like a 10,000 base pair stretch at 30% difference, which means there are going to be 3,000 BAM annotations for each overlap, okay? And then there's 2 billion of them, okay? And you have to encode them all, right? So BAM is a crap standard, okay? It's good for Illumina. It's good for high quality data. It's not good for this data, okay? Well, okay, so I could go the other way then, say, well, I'm just going to record the overlap, and later on, if I actually need the alignment, I'll compute it on the fly. I'll just compute it. But that requires a full Smith-Waterman alignment, right, which takes n squared time. Now, if this is a 10,000 base pair alignment, right, that's 10,000 squared, right? That's getting pretty heavy. So that's not good either, right? So what am I going to do? I need the alignment. Well, and I was really working on this problem. So here's the idea that I came up with, which is what I'm going to do is every 100 base pairs in the A read, I'm going to put down a line, okay? And I'm going to say for every 100 base pair segment of the A read, I want to know where that path passed through you, okay? So I'm going to keep those purple points there, right? So for every 100 base pairs, it's like I'm going to know this 100 base pairs aligned with 88 base pairs in B, and this 100 base pairs aligned with the next 97 base pairs of B, okay, of the read. So I'm going to basically keep these trace points, which in the graph corresponds to those points, and I'm just going to keep track of the B coordinate. Now, I don't need to keep the A coordinate, right, because I know I did it every 100th, right? So given A and AB, I know exactly where the intervals are, okay? But, and so what's really cool is that now if I want to give you the alignment, if I repeat that information, right, which is 1/100th of the amount I would need in the BAM style, right, then I can get back those alignments in n delta time by simply doing dynamic programming between the points, right? But the points are spaced 100 base pairs apart. So that's a fixed size, right? It's a 100 by 100 problem, right? And then there's n over 100 of them, right? So that's n times 100, right? So it's a compromise. So I can pick delta to be any value. I can pick delta to be 50. I can pick it to be 200. So I basically have a knob I can turn that gives me any space-time ratio I want, right? I can have it be more space efficient and take longer to deliver alignments, or I can have it be less space efficient and deliver the alignments faster, but I have a knob, right, that gives me a compromise, okay? And so that was very cool, but here's the thing that's really important to scrubbing. It's like, why am I doing this? Because I'm supposedly talking about scrubbing and quality values. Well, what I'm going to do, okay, this was the cool part. This was the part where you know, I wanted to run down the street naked, okay, which is that between each one of those points, right, I have those points. What I'm going to do is, in addition to keeping track of the B spacing, I'm just going to keep track of how many differences there were between those two trace points, right? So for each 100 base pairs, I'm going to know how well it aligned with the B read. You know, were there 3 errors in that part of the alignment? Were there 5? What were there? And that's 1 extra byte, okay? So including the difference information, this is 30 times more efficient, okay, than a BAM implementation, okay, already, all right? For delta equal 100, okay? But now let's come back to this pile. Okay, so now I've got all the alignments with B, okay? But for every 100 base pairs of A, I know how well the B read aligns with it, right? And I'm going to color it according to that. Okay, so where purple means bad and white means good, right? So purple alignment is a bad alignment. Well, right, it's like suddenly somebody turned on the light, right? It's immediately clear, right, that this part of the read is one of those regions where things went south, right? Because everybody says you're bad, right? If I'm a good piece of data, right, if I'm a good 100 base pairs, then certainly in the data set there should be some other guys that say, I like you. You know, there should be, you know, if I did 100x sequencing, I expect to at least find 40 or 50, maybe even 100 or 150. They'd all score with me and say, yeah, you're 15% or you're 12%, right? But no, everybody says you're crap. I just can't agree with you, okay? Well, then you're bad, okay? Here's another region where it's very hot, okay? And sorry, I'm not sure what I said, but glad you liked it, Richard. Okay, so there you go. So you just do a simple voting, okay? And you can see now what I've done is I've colored the A read. So suddenly I have a quality value for every every 100 base pairs of the read, right? It's unlike other technologies, I don't want to know the quality of every base. I just want to know, is this a good stretch or a bad stretch, right? I want to have an idea of what that curve is. And so at a resolution of 100 base pairs is enough. And what's really cool about it is it's completely intrinsic. I don't need anything from the machine. I just need the data set, right? So I call it an intrinsic quality value because it just comes from the data set, right? So in some sense, it says that really, as long as we do enough shotgun sequencing, we never needed quality values, ever, okay? They're not necessary. You can do this on Sanger data or on Illumina data if you wanted to, okay? And so now that I've got this kind of stuff, I can really go to town, but I want to finish up. So I then took this idea and I built a really cool scrubber. So I got these intrinsic quality values. Now I have an idea of every 100 base pair stretch if it's good or bad. I basically kind of decided that there was going to be good stuff that was definitely good, stuff that was definitely bad, and stuff in between. It's kind of one of my other things that I like. Most problems, there are things that are clearly good, there are things that are clearly bad, and you spend all your time on the yellow stuff in the middle, right? So I said, okay, you tell the scrubber what's definitely good, you tell it what's definitely bad, and it'll work on what's in between, okay? And then I do a bunch of things to remove all the switches, and I think I'm just gonna skip over it because I need to conclude. Again, I had a lot of fun with the details and, kind of figuring out what all the patterns were. Very interesting stuff. Let me just kind of give you maybe the overview, which is that what's interesting with PacBio data is that you need to, basically about 19% of it should be thrown away. It's just too noisy, okay? It's just bad. The company guys aren't too happy about that, but frankly, I'm really happy with that. I mean, 81% of the data is still good, okay, and absolutely usable. And so I repair I break the chimers, I repair if there were just really bad stretches that were actually just bad stretches, I repair them. And the interesting kind of idea that I came up with is I do what I call patching. So oftentimes if a segment of a read is bad, what I do is rather than kind of break it into two pieces, what I do is I take one of the B reads, I look at all the B reads and I find the best one, right? And I take the sequence from that B read and I patch it in. Okay, I just put it in place, so I completely substitute it out. I end up patching about 4% of the data, 4%, which means effectively that in any voting strategy, the patch data is never going to overwhelm the real data. Okay? So it's a perfect thing, but the nice thing is that I keep the long reads, right? I don't want to break a read if I don't have to. If I can keep that thing together, and in fact, what you'll see is in a 20,000 base pair read, there will be one of these holes every 10,000 base pairs. So if you don't patch, you're going to substantially reduce the longevity of your reads, right? Their length. But here's the cool thing. Okay, which is this is what it looks like before, okay, and this is what it looks like after, right? Quite a bit nicer, okay. But here's even cooler, okay. One more thing. I'm almost finished, Carl. Sorry. So there are two ways to use the scrubber, to use this tool, okay. One of them is you can just scrub the data and then run the rest of the pipeline, okay? And for example, we did that on the human 84X chromosome data set. It immediately improved your assembly. So I'm about to release the scrubber. I hope to release it this summer. And if you're somebody who's using this, then the scrubber is going to be useful, right? Because even if you want to run the rest of the pipeline, if you just scrub before you do everything else, you're going to get a better result. It's like a win-win, right? Just scrub whenever possible, okay? But there's another cool way to use this, which is to basically get rid of the corrector, right? To get rid of Jason's corrector altogether. Because if I've scrubbed the data and I've actually guaranteed that it's good, then there's no point in doing that correction anymore. I can go right to assembly, right? So that's the way to do it, okay? I call that correction-free assembly, okay? And basically, I then said about the following thing, which is I took a 30X E. coli data set because I was interested in the thing. I threw 20% of it away. Okay, and then I took K12, right, which is the reference, and I mapped reads to K12, and here they are mapped, okay? And this is a section of K12. Here's a repetitive element. But you can see it's very, very neat. I mean, you can see a little bit of yellow, I think, if your eyes are good. So in other words, these guys are white not because they didn't color the quality values, but because the quality values are all good. They're all matching at 15%, right, which is cool, okay? And then here's everything mapped to K12, okay? And basically, K12 is covered. And basically 99.3% of the scrub reads map end-to-end against K12, right? So 0.7 of them still have an artifact. But here's the cooler thing, which is I did assembly on these corrected reads, So no correction, no nothing, just kind of threw it into a string graph or Solaris-style assembler. I get, right, I mean, I told you, if you scrub, you get perfect assembly. You scrub, you get perfect assembly. Okay, so there's the evidence, okay? So let me tell you about, I guess, one other thing, which is repeat. So there's one other thing that people haven't really addressed in assembly. Like I said, I mean, the problem is not to improve the thing that puts the pieces together. The problem was to scrub the data, okay, and that's been done. And the other problem is really to deal with the repeats. But the way to deal with the repeats is not in the assembly. It's too late. The time to understand the repeats is before you do the assembly. In other words, what I'm proposing and what I'm building today is a tool that will find all the repetitive elements before I even try to put the pieces. Because I will know what all the elements are. Okay, and how can I do this? Well, it's really simple. I take 1% of the data set and I compare it against the other 1% of the data set, which is 1/10,000th of the total job and 1/10,000th of the total CPU hours. And when I do that, I get something like this for Arabidopsis. Okay? So you can see, there's a lot of reads that are clearly, you know, they're single copy, but some of these reads, I mean, 1% of the data is like something blew up, right? Okay, and what's going on, as it turns out, is that there's 60,000x of the chloroplast in there, okay? And if you take a look at those reads that have those big pileups, they're all bits of the chloroplast, right? So it's clear I can pull out the chloroplast, right, just by doing a 1% to 1%. Here's melanogaster, okay? All right, what are those pileups? Those are retrotransposons, right? I can see them, I pull them out, I map them, okay? So what I'm gonna basically do is I'm basically, and it turns out that 95% of the overlaps that you compute for the fly are repeat induced. So the goal then really is kind of using this idea is to actually get the signature of all the repeats, right? They show up obviously statistically because read sampling is Poisson, and then analyze those pileups and look at those repetitive elements to build models of them. And if I do that and I build the models and I soft mask the models, the overlap, by the way, will go 20 times faster because I'm going to compute 1/20th as many overlaps. Okay, so to conclude this talk, and I thank you for your patience, in principle, most important thing is that perfect assembly is in your future. Okay, I'm pretty convinced of it, okay? I built some modules. I've got this idea of trace point, intrinsic quality values, so it's kind of geeky stuff. It's kind of fun, but I think is moving things forward. And then I built this scrubber. And what's forthcoming then is this repeat detector and modeling idea, which is probably going to take us a year to do at least, which I think is going to then again take us to the final conclusion. Thank you very much.